Today I will tell you about our recent work on microRNA like small RNAs in Plasmodium falciparum. As you may know, Plasmodium is the parasite responsible for malaria, and increased drug resistance of the parasite, as well as insecticide resistance of the mosquito vector, has generated a need for novel approaches to combating malaria. In particular, we're searching for a small RNA-based gene regulatory pathway which can regulate gene expression at the transcriptional DNA level or the post-transcriptional RNA level. This is something that has not yet been observed in Plasmodium. The phenomenon of double-stranded RNA-mediated gene silencing, or RNAi, in the Plasmodium parasite had been reported in several studies. Moreover, tightly controlled transcription of genes in the blood-stage malaria parasites, despite a limited number of available transcription factors, hints that small RNAs could be involved in gene regulation. Although there are double-stranded RNA silencing effects, none of the core protein components are identified which can process the double-stranded RNA in Plasmodium. This is a mystery we're trying to solve. However, previous bioinformatics searches and wet lab experiments failed to show presence of microRNA and the requisite proteins for double-stranded RNA silencing in Plasmodium. I will start by reacquainting you with microRNA, SNO-RNA, mertrons, and by giving you our minimalist model of microRNA regulation in Plasmodium. Our theoretical search consists of a bidirectional microRNA search algorithm, as well as a protein homology approach. Finally, I will list our planned experiments. Let me take a moment to refresh your memory on the classical microRNA gene regulatory pathway. First, a microRNA hairpin, or primary microRNA, is transcribed from the genome. A protein drosha then cleaves this microRNA down to a smaller hairpin, precursor microRNA. This is then exported from the nucleus, where dicer binds the hairpin and cuts off the loop. The microRNA and its complement are then free to dissociate, and argonaute binds the mature microRNA in an RNA-induced silencing complex, which is free to find targets on the messenger RNA, usually on the 3' untranslated region, which blocks translation or marks the messenger RNA for degradation. An interesting new microRNA regulatory pathway was recently found in Giardia, a close relative of Plasmodium. These microRNA were located in the small nucleolar RNA, or SNO-RNA, of Giardia, and their processing pathway involved simply exporting the SNO-RNA hairpins from the nucleus, where argonaute and dicer would then take over in the classical microRNA processing pathway previously described. In another instance, a non-canonical pathway of microRNA processing can also occur where the dicer-mediated cleavage can be bypassed to perform transcriptional silencing, also called epigenetic silencing, at the DNA level. This happens by DNA methylation, which depends on the ratio of the microRNA and its target RNA while bypassing the dicer-dependent dsRNA cleavage. So far, while SNO-RNA have been identified in Plasmodium, none of the required proteins, argonaute or dicer, are known. However, if we could find microRNA originating in Plasmodium's SNO-RNA with targets on virulence genes, it could yield means to control the progression of malaria. A mertron is one modification of the traditional story of microRNA gene regulation. If you haven't heard of it before, it's a contraction of microRNA and intron. In particular, the microRNA is coded in an intron. This bypasses the need for drosha, since when the intron is removed from a newly transcribed messenger RNA, a debranching enzyme can process this RNA lariat into a precursor microRNA for us. However, so far this has not bypassed the need for dicer or argonaute, but can help us overcome the apparent absence of drosha in Plasmodium. While not strictly necessary, SNO-RNA can indeed originate from introns and thus possibly exploit this pathway. A side note, mertrons have been found in fruit flies, a little worm named C. elegans, and humans. A SNO-RNA, or a small nucleolar RNA, is a type of RNA that typically modifies ribosomal or spliceosomal RNA to provide structural stability. SNO-RNAs can also generate microRNA-like small RNAs by secondary processing, which may or may not require the help of RNA lariat debranching enzyme. Our model is based off of the mertron model, originating from SNO-RNA, which may end either in double-stranded RNA cleavage and RNA silencing by a non-classical pathway, such as regulation directly on the DNA, though we haven't ruled out the idea of regulation on the messenger RNA at the time of translation, as in the classical microRNA pathway. Snow RNA-derived microRNAs are not necessarily mertrons, but there is not enough data to rule out the possibility of the involvement of the lariat debranching enzyme in this case. Either through processing by a debranching enzyme or through direct folding, a snow RNA may then end up as pre-microRNA, which by some yet unidentified protein must end in a mature microRNA. In an absolutely minimalist scenario, a yet unidentified Argonaut protein may recognize a target on a newly transcribed RNA and recruit a second protein to directly methylate the DNA, affecting downregulation of a gene. This model avoids the need for dicer androsia, but still requires an Argonaut analog. Certainly, other researchers have searched for microRNA regulation in Plasmodium in the past. Cloning studies have attempted to find microRNA specific to Plasmodium without success, and searches for dicer and Argonaut proteins in Plasmodium have also come up empty-handed, including our own search. However, these studies have not looked into the intron-generated small RNAs, despite the fact that more than 50% of the genes in Plasmodium contain introns. We hope that our hypothetical model avoids the problem of lacking some of the requisite proteins in the traditional model, and that previous microRNA were missed as part of snow RNA. For this talk, I will mostly focus on putative snow RNA-derived small RNAs that could perform microRNA-like functions. In our search, we used some newly available data, namely a database of 3' untranslated regions available from studies of ESTs, and a validated list of snow RNAs in Plasmodium. We also speculate that it may be possible for dicer or Argonaut to be picked up from a host cell. Our search algorithm is unique in that we lack both knowledge of the microRNA as well as their targets. In order to overcome this, we try each possible microRNA originating from the snow RNA against each 3' untranslated region, and then propose the snow RNAs with the best hits should be our initial guesses. Thus, our bidirectional search starts with the sliding window approach, taking each 21 nucleotide group from each snow RNA. From this, we record the strongest binding duplex that could be formed from each 3' untranslated region and that hypothetical microRNA. We include no notion of a seed region and use only the physical energy parameter. We then filter out weakly binding pairs, less than 20 kcal per mole, and poorly conserved pairs, using 90% as a threshold of conservation. The hypothetical microRNAs with the most hits after these filters are then our best guesses as to what might be a real microRNA. We were happy to see that our results tended to be near the base of hairpins in the snow RNA, which would be the likely location if a dicer or dicer-like protein were to trim the hairpin. Highlighted on this first slide is a target on a cytoadherence protein, which is exactly the type of target affecting virulence we would like to see. On each of these slides, our putative microRNA is highlighted in blue on the figure. The results of this are some interesting hits, but we have nothing conclusive yet. We would also like to perform our own search for dicer or Argonaut proteins in Plasmodium. These motifs are so broadly represented among different organisms that we employ a new tactic to attempt to find even vaguely resembling them. Owing to the evolutionary diversity of these proteins, we select against all known motifs from PFAM, then run NCBI BLAST-P for each of these known motifs in different organisms against each translated protein in Plasmodium and record the most unlikely results, as measured by an expectation value. This program is quite fast and runs within a day or so, so we could also use it to search for analogs of any protein domain you can find in PFAM.